Faculty of Earth Science have been established in 2012. We are in one of the youngest university in Malaysia. We have a vision to become a pioneer in sustainability, environment, and natural resource through entrepreneurship agenda.
Currently, Faculty of Earth Science offering 3 undergraduate programs. We also have 2 preparatory programs which managed at Faculty of Earth Science.
For Bachelor of Applied Science, Natural Resource with Honors, the focus of this program are much more on our biosphere, which means we are focusing on our plants, our animal, and also the management of our tropical forest. For another program, which is Bachelor of Applied Science, Sustainable Science with Honors, the program itself relate more on environmental element, which is we would like to sustain our environment together with the importance of economy and also social.
Always apply teaching and learning section by using technology such as by using GIS, drone, geospatial mapping technology, and also modeling. Students also be taught with to integrate geoscience understanding from classroom or practical learning with geoscience data from a variety of sources, creatively using the latest digital technology.
Science Preparatory Department is a new department and need to taking care and manage of 2 academic programs. Both of these programs will need to learn biology, mathematics, chemistry, and also physics.
Faculty of Earth Science Museum, actually we have various dry and wet specimens such as insects, small mammals, reptiles. Besides that, we also have rocks, minerals, and also fossil. So actually all those specimens being collected by our students.
To stay relevant, you have to be flexible. Besides flexible, we have to be unique. In Faculty of Earth Science, we have multidisciplinary research. Recently, we are focusing on the research on minerals and the fossil. Other than that, we use remote sensing and GIS in monitoring forests and urban planning. This research has been successfully done within our capacity of staff, supported with the grant that we received from government and other agencies.
We are optimistic to become one of the best faculty that focus on in environmental in Malaysia.
I'm Siti Aisyah Nawawi, a moderator for today's webinar. A very good day, Assalamualaikum, and salam sejahtera to invited speaker, viewers through Facebook University Malaysia Kelantan Rasmi and YouTube UMK TV. Welcome to the invited speakers on FSB International Webinar organized by Faculty of Earth Science, University Malaysia Kelantan. To our FB Live, University Malaysia Kelantan Rasmi and YouTube viewers, if you are watching from Malaysia or other countries, do not forget to leave a comment or questions. Today, this FSB International Webinar is about natural drug research and development during COVID-19 pandemic era. COVID-19, a disease induced by SARS-CoV-2, which is severe acute respiratory syndrome coronavirus 2, has been the cause of a worldwide pandemic. Now, we are here to visually listen and learn from our expert how the research and development on natural drug during pandemic era. Before the talk begins, allow me to introduce the speakers. Our first speaker is Dr. Sudeshna Data. She received the PhD degree from the University of Calcutta and did her PhD on application of nanoparticles in electrochemical sensor. Her research interests includes synthesis and characterization of metal nanoparticles and their potential application in the electrochemical sensor for medical, food, and environmental purpose. She has 12 years of research experience and published a number of papers in international journals. Her areas research interests include designing and application of electrochemical sensor and designing drug delivery vehicles using phytochemical and probiotics. Without further ado, I would like to welcome Dr. Sudeshna Data to deliver her talk on conjugating curcumin to water-soluble biopolymer via pH-responsive succinate linker for improved stability and sustained release. All right, Dr. Sudeshna Data, the platform is yours.
Thank you so much, Aisha, for the kind introduction. And thank you, the University of Malaysia, Kelantan, for giving me an opportunity to deliver my, to share my research experience with a broader audience. So without further delay, I would like to start my talk. Just a minute, I'm new to this. Is everything fine? Can I start my presentation? Here is my topic of my presentation is conjugating curcumin to water-soluble biopolymer via pH-responsive succinate linker for improved stability and sustained release. I have done this work in IIT Kharagpur, and Professor Ramkrishna Sen of Department of Biotechnology has supervised my work. Now, as we all...
I'm sorry, Dr. Sudeshna, for interrupting. Yeah. We can't see your slide, sorry.
Sorry, but what to do? Then stop sharing. What to do then? Sorry. Share.
Yeah.
Wait, if I know there's nothing share. Just give me a few minutes.
Okay.
Share screen. This one share. Can you see now?
Okay, yes, we can see your slide now.
Yeah. Thank you. Can I start again?
Yes, you can. You may start again.
Thank you. Sorry for the interruption. The topic of my today's talk is conjugating curcumin to water-soluble biopolymer via pH-responsive succinate acid linker for improved stability and sustained release. I have done this work in IIT Kharagpur, and Professor Ramkrishna Sen of Department of Biotechnology has supervised my work. Now, as we all know, curcumin is a very known nutraceuticals. It has got many, and it's a part of our daily life, as it has a potential to stop many life-threatening diseases. Though it is a spice that is present in turmeric, but and it has got many properties like anti-cancerous, antioxidant, anti-inflammatory, and many more. But there is some properties of curcumin made it not a very friendly material for clinical use, because of it is highly insoluble in water, and it lost bioactive structure as soon as it get inside the human body, and it has got some unwanted side effects like constipation, vomiting, nausea for more use of curcumin. So delivery of curcumin inside human body is a challenging task. Now we have adopted many strategies to address these challenges. We have chosen a biopolymer, that is exopolysaccharide from Bacillus megaterium RB05, and we have used this biopolymer as a vehicle to deliver curcumin inside human body. And for that, we have synthesized, modified, and characterized the polymer-based conjugate, and also enormously studied the drug release and stability of the conjugate. Now, here is the objective of my work. We would like to investigate the naturally occurring water-soluble polysaccharides from Bacillus megaterium RK05 to overcome the drawbacks that prevent curcumin from being translated to clinical use. Now, why we choose this Bacillus megaterium, which is a gram-negative bacteria, because it is an unique bacteria which is isolated from our lab, and its anti-cancerous property was already established and published. So we choose the exopolysaccharides coming from this bacillus to use as a delivery vehicle. We have also studied the properties and morphology of synthesized polymeric system using FTIR, MALDI-TOF, SAME, FE, SAME, NMR, etc. And we have also explored the conjugate we prepared with this drug, with drug means curcumin with biopolymer, and we have also studied the in vitro drug delivery and blood compatibility of the conjugate. Now coming to the process flow diagram of EPS production, which is the first step of my work. We have taken the Bacillus megaterium RB05 and grew this in a general minimum salt media, and then centrifuge and take the extracellular material, and then we did the standard procedure like acetone precipitation to remove all the proteins, and then dialyzed and then lyophilized exopolysaccharide, that biopolymer we used for conjugate preparation. And in second step, we have modified the curcumin in such a way with coupling reaction so that it binds with the polymer. For the coupling reaction, we did a chemical DCC DMAP coupling reaction, and for each step we have checked with thin layer chromatography whether product is present or not. And after that, when a violet spots is obtained, which indicate that product is formed, and then we have run the column purification to purify our product, and finally we took the modified product, which is nothing but from curcumin to curcumin hemisuccinate. Now from the curcumin hemisuccinate, we bind that with our polymer. That is the final step. Then we did another coupling reaction, and from that curcumin hemisuccinate bind with the exopolysaccharides. In this way, we have modified the curcumin in such a way so that it can bind with the biopolymer, and it can be used as a conjugate for curcumin delivery. Now here are the results. In first picture, you can see the bacteria with exopolysaccharides. As you can see that exopolysaccharides are covering all the outer shell of the bacteria. And in the second picture, you can see the only exopolysaccharide, that is the biopolymer, which is like very round, irregular in shape, and it is globular in structure so that it can hold water. As the biopolymer structure of the exopolysaccharide contains many OH group, so that it can hold lot of water in it. And finally, this is the structure of EPS curcumin conjugate. As you can see, this is different from the other structure, which confirms the formation of EPS curcumin conjugate. Now coming to the FTIR studies. We have done FTIR in each step, and the final step, which is we have done only exopolysaccharide, that is the biopolymer, only curcumin, and the conjugate also we have done the FTIR analysis. The final curcumin EPS FTIR has both the spectrum from curcumin as well as from the biopolymer. And as you can see that there are characteristics bonds present, like from this band, say from 1100 to 1000 centimeters is due to the vibration of glycosidic bonds, and this is the 1730 is a unique bond which is like due to the succinic acid linkage, which confirms that curcumin is successfully bound to the EPS backbone. And here is the stretching peak of 2882 due to the CH aliphatic stretching peak. And these are there is the high curve here in between 3396 to 3350 because of the OH stretching. And as you can see, that curve is quite high because many OH group is present inside the EPS structure. Now coming to the NMR spectra. NMR spectra we have done to confirm our product. For curcumin, now as you can see in curcumin structure, there is two OCH3 group, and this will give a typical peak around 3 point something, and this is the pure curcumin, and you can see this OCH3 peak is observed. And in EPS, that is exopolysaccharide biopolymer, you can see lots of small peaks due to the proton of exopolysaccharide, and here is the alkali region. In case of curcumin hemisuccinate, you can see CAH group, which is modified due to the succinic acid linkage. And in case of curcumin hemisuccinate and polymer complex, there is COOH group is missing because it is modified due to the binding with the OH group of biopolymer. This NMR results confirm that curcumin is successfully inside the biopolymer. Here is the Marlitov results, which confirm all the monosaccharides presence in the exopolysaccharide, and also confirm the conjugate. And this biopolymer is around 170 kilodalton in size, and it has got many monosaccharides, right, like glucose, sucrose, fucose as its monomers. Now coming to the DLS studies, after the only exopolysaccharides are having the round in shape and around 180 nanometer in size, but when curcumin EPS is formed, the structure increased and the size of the particles has also increased. It's around 290 nm now. And the zeta potential is very important to know the surface charge of the particle, so that it will not be repelled inside the human body. And the EPS curcumin conjugate is negatively charged, and it has got zeta potential around minus 40, which is very stable. Now here is the drug loading capacity. As I told already, that curcumin is insoluble in water, but when EPS curcumin conjugate is formed, its solubility increases, and this is how curcumin can be delivered inside the human body and overcoming the obstacles. Around 3 mg of curcumin EPS was able to solubilize in 1 ml of water, and this corresponds to 990 microgram of curcumin. Now coming to the another result, that is the stability of curcumin, that we have checked that the conjugate is very much stable in phosphate buffer salines, and we checked in 1 hour intervals for around 8 hours to check its stability. And it also confirmed that generally curcumin undergoes the rapid hydrolytic degradation into vanillin and ferulinic acid above neutral pH, but due to its assembly with biopolymer, its hydrophobic curcumin came into the miscel core, and the hydrophilic chain of EPS is surrounded it, so its solubility increases, preventing its rapid degradation inside the human body. Here is the results of the release structure of curcumin. That is, we have also studied the release of curcumin from the conjugate in 2 different pH. One is 7.4, which is physiological pH. Another pH is 4.5. We have chosen this pH 4.5 because the tumor cells are slightly acidic. So we have to check that our curcumin should release at a particular pH, not at physiological pH. And that's why we have studied the release of curcumin from the conjugate in both the pH, and we have obtained the results which shows that due to the succinic acid linkage, which is a pH sensitive linker, curcumin can be released from the conjugate at 4.5 in a better way than physiological pH. This results also indicates that this conjugate has high potential to use as a anti-cancer drug delivery system. Now, to summarize my talk, I would like to say that conjugations of hydrophobic curcumin to hydrophilic backbone of biopolymer via succinic acid spacer was successfully done, and with all the results from FTIR, SAME, MultiTOF, and DLS and DETA studies, we can see that the conjugate is now negligibly susceptible to degradation, and its aqueous solubility has enhanced. And this micelle showed the rapid release of curcumin at acidic pH compared to the physiological pH, which is 7.4. In vitro release studies also confirm that pH sensitivity release of curcumin, which is conducive to the tumor microenvironment. Generally, the tumor environments are acidic in nature. So, if our curcumin can be delivered in a acidic environment, then we can target more target specific delivery of curcumin into the tumor cells, and then it can kill the cancerous cells. And this micelle has the ability to improve the time of systemic circulation with enhance the EPR effect owing to its negative zeta potential. And finally, here is some of the references of my work. And still we are doing the anti-cancer test, and cytotoxicity test is currently going on. And thank you very much for your time and patience, and thank you very much to the University of Malaysia, Kelantan, for giving me an opportunity to share my research work. Thank you very much.
Okay, thank you very much, Dr. Sudeshna Data, for very interesting presentation and informative sharing knowledge in conjugating curcumin to water-soluble biopolymer via pH-responsive succinate linker for improved stability and sustained release. This study could be applied by other researchers as curcumin is a natural product with immense medical assets. Thank you very much again, Dr. Sudeshna Data.
Thank you so much.
Okay, now I would like to introduce our second speaker, Dr. Suganti Apollosami. Yeah, Dr. Suganti. Yeah. She was born in Perak. Her first degree in resource biotechnology in UNIMAS, PhD in plant biotechnology USM. She is a senior lecturer in Faculty of Earth Science UMK and research fellow in Institute of Food Security and Sustainable Agriculture. She was working on various termites for new termiticide discovery and wild changes for insecticide, immune system booster, and as functional food. I would like to welcome Dr. Suganti Apollosami to deliver her talk on new perspectives on how to discover drugs from medical medicines. All right, Dr. Suganti, the platform is all yours.
Thank you, Dr. Moderator. Thank you, Dr. Aisha, for the very warm welcome, and also to FSB for the opportunity. For that, let me share my screen.
Yes.
All right. So I guess it is quite clear, Doctor. Is it okay? All right. So hello everybody. My name is Suganti Apollosami. I am from Faculty of Earth Science. And thank you very much for the opportunity once again. I cannot thank enough. So the title of my talk today will be Discovery of New Drugs from Herbal Medicine, a New Perspective. So when we are, facing this era, the pandemic era, a lot of us have been asking ourselves, is there any cure for this coronavirus? Can we go back to our traditional knowledge, the herbs, can we find any kind of cure or any kind of medicines using the herbs that was left behind by our ancestors, and also our, the knowledge that have been passed down generation to generation by our family. So when we talk about the discovery of drug and all that, people, the layman, the common people, we sometimes don't have any idea what goes behind, behind the screen, behind, the experiment, and also the steps involved in drug discovery. So for that, maybe I can explain a little bit, and also we'll be discussing more on what happens, the process, from the herbs and also the extraction until the drug discovery, and then the challenges we face in this in this field especially. So of course, we have to go back to the basics. What is herbs? Okay, so herbs is defined as a plant or plant part used for its scent, flavor, or therapeutic properties. So in terms of herbs medicine or herbalism, what is this? So according to World Health Organization, it is defined as plant-derived material or products with therapeutic or other human health benefits containing either raw or processed ingredients from one or more plants. So I'm very sure a lot of us have this experience. If let's say we are still, with our grandparents, or we have the older relatives, when we complain we are having headache, or we have, stomach cramp, they will be definitely recommending us, oh, let's drink, I'll make you fenugreek water, or drink this with these leaves and all that, or I can make you some jamu. Malaysia, they have this jamu and all that. So these are all categorized under herbal medicine. So it has been practiced in within the unit, the family unit, and it has been passed on from one generation to other generation. Sometimes it is not documented, and most of the times it is documented. What is the history of this herbalism or herbal medicine? So when we look back at the history, and then we find out that it has been here for a very long time. It's not like 100 years old traditional knowledge. No, it's not. It actually have a history of thousands of years ago. The first one was documented from the Aztec community, where they have their own Aztec medicine, right? So the doctors, they have documented about 1,000 medicines from the plant, and it all has been done through observation, surveys, and also, based on their clinical data, when they give some extracts to their patient, and then they actually observe what happens to their patient, and then they jot down. The main thing that we have to be thankful is that they documented it, and until now we can still access what is being documented. So that is in within Aztec community. So Aztec community, what is that? Okay, they are a community that can be found in from middle of Mexico. All right. So what about other communities or other places? Do they have this? Do they practice this herbalism? Yes, of course. Where there have been documents found from ancient Greeks and Romans, who are very well known for their herbal medicine use. Greek surgeons, they have been reported to use herbal medicines, and then they also have their own, Materia Medica texts, which actually exist like 1,500 years ago, and very well documents of the usage of certain plant materials that can be used as medicines, and to alleviate fevers and all that. Right. So there are also documents found from ancient Egypt, from ancient Indian, and also the famous Chinese community as well. So all those we are still able to refer them. We are still able to access them because of their documentation. And some, of course, the plants are very much localized, meaning that the community can use whatever plants that can be found within their region or within their place, and then they document it. Of course, when we talk about plants in some other region, they cannot find it, so they have what they have there, and then they document it. So when we have this, very full or very well compiled documents, and that will become our main reference, right? For main reference for the medicine that we have today, right? So that's about the herbalism, the history of herbalism that from our ancient people and also our ancestors. So what happens now as the time progresses, right? The herbal medicine, they become to be overlooked, right? Their popularity when dimmed, with the introduction of new or modern science and also modern medicine. So there are people, they forgot the existence of this traditional knowledge, traditional method, herbal medicine, and then they shift their focus to other kind of medicines, all right? The modern, the synthetic ones, right? And after that, after 18th century, the herbal medicine, they regained their popularity, right? They regained their popularity, and now in 21st century, the modern medicine is actually much more focused on discovery or rediscovery of drugs from our herbal medicines, all right? So this is supported by an array of research done in this field, using the plants and also the documented traditional knowledge, right? The documented traditional knowledge to rediscover the medicines that can be useful to humans, right? So what happens next is that there have been a lot of publications happening, right? There have been a lot of publications that reports, the finding of new drugs and the useful drugs, all from this herbal medicine. And these are some of a very good journal which can be accessed freely. And then all reporting on this, folk medicine, right? And also some other traditional medicine that have been previously used, freely within our families, and now they actually have this recognition, worldwide recognition, and can be used as a drug, to cure certain sickness and illness, right? So these are some of the papers that actually targeting more on the coronavirus, right? So this is also the evaluation of medicinal herb as potential therapeutic option. So people are no more, depend solely on the modern medicine. They are also, recognizing, they have started to recognize and started to use the herbal medicines as an alternative medicine, right? So this is one of the progress that we are facing now. And then the finding of this drugs and all that, okay, it has been going on for a few years, few decades already, right? And some of us don't even realize that what we are consuming as medicines, actually they have originated from herbs and plants, some of us don't even know what we are consuming, but the supplements we take, where are they from? We don't even know that, isn't it? Yeah. So for example, the aspirin, the source plant is from Salix species, right? The atropine, which can be used, to treat cardiovascular, anticholinergic, hyperdilation, all that is actually from our plants that can be found around us, right? So when we look at the trend, right, based on the survey data, right, people are more, they are becoming more associated with this herbs, herbs related and botany related plants, medicines, right? And in 2000, all right, in 2020 until 2030, all right, they predicted the nutraceutical market will actually grow and continue to grow, all right. So the market size will become very big, all right, and it is an opportunity, all right, for researchers, all right, who are very much into this drug discovery field, all right, to work on it and then to find new medicines, all right, all originated from herbs, all right. So let's look at the US herbal supplement market size. So why did I choose this US market, not other country for an example, right? So maybe most of us didn't know that, but US, the people in US, they are one of the highest consumers for herbal or herb-based medicine, right? Right. So they actually have the biggest market, yeah, for this herbal supplement and also nutraceuticals, right? And when you can see the demand for supplements and medicines based from ginseng, ginger, turmeric, flaxseed, echinacea, and also moringa, they are growing steadily, right? There is no sign of decrease, right? So this is the market size in US alone, right? And then let's look at the market size by region, right? So as you can see, the demand for herbs, right, and medicines from herbs, right, is actually growing very steadily. Not only in America, but also in Asia Pacific, Middle East, Africa, Latin America, right? Okay. So what about the the supplier or the drug company, right, who produces, who does the research and also produces this drug, right? And they also reported, to see a very steady trend, the trend, growing trend actually, in terms of the drug market, right? So the top companies, right, Abbott Laboratories, Bionova, Eli Lilly company, they are all have patented few drugs every year, no less than 10 patents for drug formulations alone every year. And how much they spend for each drug discovery? Billions, right? Billions of dollars, right? So it is actually is driven by the market and the demand and also the supply. All right. So when we have looked into the market, all right, we have looked into the demand, all right, the top companies that produces this drug based on herbs, right? So let's look at, quickly look, right, what actually involved in this drug discovery, all right? So this is a very simple, figure, right, that actually shows you the difference, right, between the traditional system of medicine. So what is involved, right? So usually, people who actually practices this traditional system of medicine, they have this, they will be doing the preventive measure, give some medicine made of herbs, right? And then they will look at observe their patient first. Then after that, they will go into the curative medicine, then alternative measures, and finally the surgery, of course, right? So these are the steps that are involved in the traditional system of medicine. So when we compare that with our allopathic system, the one that what we call as modern medicine, right? So it actually has more steps, the medicines, right? And then the diagnostic techniques, and then supporting measures, observations, and then more diagnostic, then special treatment technique, and finally we have this surgery. So the medicine, right? The medicine here is actually, when we talk about the allopathic system, it comes first, right? But then for the traditional system, it has been like, we have the preventive measure, also medicine, then we have the curative medicine, and then we have the supplements to sustain the health, right? So these are the differences. When we fall sick, we go see a doctor, and then he'll be prescribing us with some medicine, and then when we are okay, we have feeling better, then we stop taking the medicines. All right, so that is the allopathic system. So then this has been rebutted by the traditional system. We said medicine means we have to eat to feel better and to sustain your health, right? And this has been agreed, right? This has been agreed by current medical field, right? And also the experts, right? So now, on your left-hand side, we have this very complicated figure, isn't it? Yes. Okay. So that is actually to show, right, the experimental phase, the exploratory study, and also the more studies, right, that is involved when we want to screen, right, screen for a compound or a chemical from herbal medicine. So we have to understand, right, when our ancestor or the traditional knowledge, right, in traditional knowledge, right, involving herbs, we do not isolate one compound, right? We actually consume them as an extract or a mixture or a concoction, right? A concoction with few other herbs, right? So sometimes it actually has a very positive effects to us. Sometimes it does not, right? Sometimes it actually give some side effects, right? So to avoid that, we are now focusing more towards the compounds, right? The compounds or what we call as the candidate compounds, right? That can actually, focus or can be directed, right? Can be directed in curing one disease. All right. Okay. So in terms of the steps involved is actually quite elaborated. Right. First involved with the biological screening, isolation of the active compound, then preclinical study. Then after that, you have the clinical trial. Then we also have to go through the the efficacy test, shelf life, and then after that we also have the post clinical and also the commercial stage, right? We have to observe still, even though it's already on the shelf, right, already being sold, but it still go through some survey and also observation for the side effects and all that. All right. So these are the the lengthy processes that are involved, usually involved. So what happens if, let's say, we don't have the previous data or the previous information on this plant? Okay, for example, we don't have any kind of documented knowledge, right, of a plant. So what will happen next? All right, so usually what researchers do, they will be screening for the compounds that present in that particular plant. All right. So when we say screen, does not mean that we wear some magical specs and then we look at them. No, it's not. Okay, we have to do a lot of chemical analysis, all right, and then we need to have the samples, enough samples ready and all that. So it actually consumes time. All right. So just for the record, all right, for a drug, for a herb plant, all right. From a herb plant, when we isolate the compound and until it becomes a drug and can be sold, in the market, it actually takes about 10 years. About 10 years, yeah, right. So it is not very easy. That's why people have been asking why there is no, medicine or drug for this sickness or illness. Why we cannot have this? We already have the illness, right? It's already killing people. Why we don't have the drug yet? It's like yesterday the illness appeared and today they want the drug. No, it is not as simple as that. All right. So the current processes, right, involved in drug discovery from herbal medicine, right? So as I told you, it involved the isolation or artificial synthesis, right? And then after that, the evaluation of safety and efficacy. Of course, we have to go through the ethical, board committee and all that, right? Then after that, evaluation by means of conventional pharmacological method, right? Without involving the human, right? This is involving the non-human, right? From pharmacological method, right? Then after that, we have to have for the regulatory approval. Then we'll be testing on humans and all that. So these are the processes, but it's not as simple as it sounds, right? Okay. So when we talk about more we go into the steps in detail, right? So these are the steps that are involved, all right, for herbs, yeah, to become a drug, right? So there are mainly 3 stages, right? Pre-drug, quasi-drug stage, and finally post-drug stage, right? So all this happens inside a laboratory. All right, so if you feel that, wow, this is so cool, I want to be a researcher, come join us in FSP. All right, okay, so so these are all the things that happens, right, in a laboratory, right? It is being done by researchers, right? So just a small, fun fact, all right? So it is estimated we have about 420,000 plant species, all right, that can be found all over the world. But until now, only 30,000 plants have been screened and used as herbs, right? Or categorized as herbs and used in the herbal medicine. All right, only 30,000. The rest, we don't have any data, we don't have any documentation, all right, to prove it have some uses and all that. So we still have a very long way to go, before we can actually screen everything. Okay, all right. So when we talk about, previously I actually mentioned we have to have like 10 years, right? To have a drug ready for selling and all that, right? So with the emerging, new technologies now, that we actually save some years. How we save some years, when with the use of latest technologies, with the computational systems, right? The computational system actually help us with this 3D structures, all right? 3D structures of the proteins, of the compounds, right? That can be found in a herb. So when we have this 3D structures and all that, we can also use the computer to give us a model, to how it behaves with the target tissues and all that. All right, what is the drug delivery form, the system that can be used without even doing it in the laboratory. We do a dry run first. All right. So if it is successful, right, with the computer, right, it is successful, then only it actually goes through the next stage, that is being screened, right, and then also in all the other steps that are involved in the wet laboratory, right? So why this is being done is actually, one, to save some years, right, and also a lot of money and effort. Right, so we are, when we have this pandemic situation where it's actually, we need the drug ready, like yesterday, not today, yesterday we need the drug ready. So everything we have to, plan our work, in regard of the time and also the money, right? So how this is being done is actually, being done with the help of these computers and all that, right? So of course, we still have to go back to the wet lab and all that, but we can save some time, right? Okay, so when we talk about, uses of herbs, you might be asking, is there any herbs, that actually successfully, turned into a drug and helped, to cure, to alleviate any sickness or any illness? Yes, we have. So this is a very, famous example, right? And I have worked with this plant, right? It is known as Artemisinin annua. So this is the project that I did for my PhD, right? So it was funded by a very big grant from World Health Organization, right? So this Artemisinin annua, right, it's actually a plant that can be found in China. So for this genus, there are about 5 or 6 species, if I'm not mistaken, right, that can be found. But only this particular species, annua, okay, Artemisinin annua, can be found growing widely in China, Nepal, Tibet, Vietnam, right, and Laos, right, have this particular compound known as artemisinin. So what is so great, what is so special about this compound? It is a compound that can alleviate and also cure malaria. So it is used as anti-malarial drug, right? So now, right, it has been successfully used to treat malaria patients, and now it is used as drug. right? And a lot of research have been going on to, produce this compound synthetically and all that, right? Another one more very famous, medicine or drug that we discovered from our herbs, right, or our traditional knowledge is the fructose skin cendrase sinensis, right? So this is a wild growing plant as well, can be found growing widely in China and also in Tibet and all that, right? So from this, we actually isolated a compound, right? And that compound is a powerful anti-inflammation and also used to treat hepatitis, right? And it can be found in the market now, right? So these are some of the success stories, right, that actually started with the help of our traditional knowledge, right? So what happens if we don't have the database, we don't have the documents, right? So we might be spending a lot of time screening, we don't even know what we are screening. And then we have to, do not only for one plant, we have to do for thousands and thousands of plants. So with the help of this traditional knowledge, we actually cut down a lot of years, right? So what happens now is that it actually shows us the importance of having a database. All right, so that's what we are doing. All right, a lot of countries are doing, right? They have their own databases, right, that stores the important proteins and compounds that can be found from any kind of herbs, all right, and what is the uses and all that. So we can actually access these databases, all right? Some of the databases we can access, and then we can have a look at the drugs, right? All of them are from our herbal medicines. Right. So now, what happens next? Right. So now we are in the era of post-genomic era. Right. So what is post-genomic era? So this is an era whereby we already know, the gene sequences of human. Right. In 2004, they already published the full gene sequences for human. Right. We have about 3 billion, nucleotides. We have 33,000 genes and all that. So we already know, what kind of sicknesses, which chromosome, holds the information for this sickness, for this illness and all that. So we already know this, right? So what happens next is that the usage of this information and to find cure, right, for certain sicknesses and all that. And by using the compounds isolated from our herbs, all right? So this is the next, all right, what's in trend now, right? With the usage of our information, the gene information, human gene information, and also to find, the compounds that can help. So the merging of these two information is actually very important, all right? And this is the current emerging, topics, right? So of course, we'll have some challenges, right? So what are the challenges? Of course, when we talk about plants, right, they grow widely. Sometimes they grow widely, right, and sometimes they are classified as endangered plants. All right, so when it is endangered plants, which means that we cannot find it a lot. We cannot be, harvesting them, right, for industrial scale and all that, right? So there must be some alternative. So what kind of alternative? We can actually use bacteria to make the active ingredient. All right, so whatever we do, right, to face the challenges, we have to be very ethical, right? As a researcher, right, I suppose everyone listening to my talk today, right, we consider ourselves as a researcher, right? So the ethics, right, we have to be very ethical, and also we have to respect the nature. That's one thing, right? So the control of the usage, right, the usage of the wild plants. Okay, now we have to control them. And we have to find an alternative, how to produce the compounds, but without damaging the nature. Right? Sometimes, the compound can be found with, can be found within the same family, right? Or if not in the species, another species of the same family, or sometimes in the same genus, right? So we can have this kind of alternatives, right? Sometimes we can even cultivate them, right? Cultivate the medicinal plants, and then produce or screen the compounds from them, rather than going back to the nature and then taking the, the samples from the nature directly. And finally, finally, we have to have documentation. Whatever we do, we need to document them and make it available freely. That is one thing that is actually a challenge that is being faced by a lot of researchers, right? We don't have the freely available data and info, right? So sharing, right? Sharing is caring, isn't it? Okay. So with that, all right, I hope whatever information that I have given is useful and can be used for your future, investigation and also research. If there is any kind of mistake, it all comes from me. And if let's say it is useful, thank God, right? So thank you again, Madam Moderator, for the opportunity.
Okay, thank you very much, Dr. Suganti, for very interesting presentation and informative sharing knowledge in new perspectives on how to discover drugs from herbal medicines. With tens of thousands of plant species on Earth, we are endowed with an enormous wealth of medicinal remedies from Mother Nature. As articulated by the ancient Chinese philosopher Lao Tzu, Mother Nature is benevolent to all living things, even a stray dog on Earth. Okay, I can see a few questions from the audience. All right, to all the viewers who are watching right now, we come to the question and answer session. Okay, we have questions for Dr. Sudeshna from Amal Najiha.
Okay.
Okay. She asked, Dr. Sudeshna, how you manage to secure and continue your research project in this pandemic era, especially in doing laboratory work? Okay.
Yeah, okay. Very relevant question, I must say. But thing is that generally our institute is closed for last 1.5 years. So basically we cannot do any lab work. So we all are doing just doing all the offline work, like writing review papers, reading more journals relevant to our research area, and just like that, and waiting for our institute to reopen, because we are many of our experiments we can only do in only in the lab. We cannot do it offline or somehow. Only some parts we are doing, like molecular docking studies, where you can computering generated, you can say how our curcumin is effective inside the and cancer cells and all those things we can do. But honestly speaking, we cannot complete our work due to this pandemic situation. We're just waiting that this COVID situation will get over and we can get inside the lab and complete our work.
Okay, thank you, Dr. Sudeshna, for your answer. Okay, we have question for Dr. Suganti. Okay, we have question from Asia Sila. Okay, the question is, there are many synthetic chemical used in place of natural chemical, good or bad? Dr. Suganti.
Okay, I would say, when we talk about, synthetic, so previously I actually mentioned about using the samples, the samples or the plants from directly from the wild, and using it for screening and all that, isn't it? So when we talk about, plants that could be endangered, right? So this is where the synthetic compounds come in, right? So it is not bad, right? We cannot have an umbrella ban, to tell that the synthetic compounds are bad. They actually offer an alternative to the natural product or natural compound. Understand? So using the synthetics, sometimes it actually reduces the use and dependency of this around on the herbs and all that. So that's why I say it is not entirely bad. All right, so it is actually good. Depends on the context of the usage.
Okay.
I hope I answered the question, right?
Okay, thank you very much, Dr. Suganti. I think we have another question for Dr. Sudeshna from the viewers here. Okay, how you evoke interest in this study, Dr. Sudeshna?
Actually, can you repeat the question once again?
How you evoke interest in this study?
Yeah, this particular topic which I have presented here, and that fascinates me because this biopolymers is a nature's gift. Like bacteria naturally produce these biopolymers. So instead of using any synthetic or chemicals for as a drug delivery vehicle, why don't we use this nature gift to deliver another nature gifts? So that's what fascinates me. And this topic was different from my PhD topics. Like I have worked with electrochemical sensors, but I would like to explore the bacteria's possibilities, and I was always fascinated by learning new things, and these things really fascinates me, whether I can use a nature's gift to deliver another nature's gift. So that's what catches my interest, and I started this research work.
Okay, thank you very much, Dr. Sudeshna. Okay, okay, we have question for Dr. Su here. Okay, in your opinion, what could we do to preserve the traditional medicine knowledge of our communities?
As I said before, documentation, right? So there are a lot of traditional knowledge that has gone unnoticed, right? Sometimes it's actually they use these plants that nobody has heard before, but it is proven to cure some sickness, some illness, some ailments within their community, right? So we do know all that. So without the proper documentation, I think the knowledge will disappear soon. When the community stop practicing them, or when we cannot find the plant at all in the wild, right? So I would say documentation is actually very important. And the second one will be educating the people, not only within the community, but also to other communities as well. The sharing of knowledge, right? Sometimes the Malay community, they have this, very precious knowledge, right? But it only practice within themselves. So it is, it is not intentionally not shared. It is unintentionally, but we can actually share them. So we have a lot of means of sharing nowadays, and I think it can be brought forward and it can be shared, right? I'm very sure researchers now, they have a very close contact with the communities. When we go for sampling and all that, they can bring back this and then they share. They share. First they share with the researchers, next share it with the other common people, all right? By writing, by videos and all that. I think this will help a lot. Yeah.
Okay, thank you, Dr. Suganti, for the brilliant answer. Okay, so I think we realized that there are many interesting questions posted by our viewers, but unfortunately, I'm afraid that all the time we have for today. I would really appreciate if Dr. Sudeshna and Dr. Suganti could answer in the comment section in the FB in any time. Okay, ladies and gentlemen, finally, we come to the end part of our session. We would like to say thanks again to the speaker, Dr. Sudeshna Data, and Dr. Suganti Apollosami, for making your time and being here for the informative and interesting talk. And to all viewers from Facebook, University Malaysia Kelantan Rasmi, and YouTube, UMK TV. Hopefully, the session will be beneficial for everybody, especially in UMK Malaysia and the rest of the world. Thank you, everyone, and hopefully we could see each other again. Assalamualaikum.
Okay. Thank you.
Bye-bye. Thank you.